So thank you very much. And can you hear me okay? Yes. Okay. So it's a treat to be here. I loved this meeting the last time, seeing the cathedral and meeting all the people here. And to those who came back again despite my talk the last time, a special thanks. And so what I'm going to talk about this time hopefully is going to be a little different than my last talk, because when I spoke before, it was more of an overview of different types of CMT. And I think that there's a sense that this is a different era that we're in now, where there really, it's gone from trying to characterize different types of CMT to really focusing on developing treatments. And I just want to try my best to try to give a sense of some of the excitement that people in the field feel about this, because I think we're really beginning to enter an era where there's really rational treatment approaches for the inherited neuropathies. So this is just a cartoon of different chromosomes. And the reason I'm showing it is just to, first of all, show an expansion in our overall knowledge of CMT. Because when I was here 3 years ago, there was probably about 40 genes known for CMT. But with the advance of new technologies and next generation sequencing, the number I'm going to show you is from just a month ago, but it's probably already out of date, that we now know of over mutations in over 82 genes that can cause CMT. And I'm not going to review the different classification systems here because I don't think I have time, but these are still subclassified into types with slow nerve conduction velocities that we call CMT1, or normal nerve conduction velocities that cause CMT type 2, or recessive forms that I get to argue with Mary about whether we call them CMT type 4 or autosomal recessive axonal forms of CMT. So in any case, there's a lot going on in terms of understanding different types of CMT. And I don't have slides of this, but I just want to try to make this point that for people who are trying to understand and develop treatments, it's really exciting when we get all these new genes, because it enables us to see in the laboratory how different proteins in myelin or nerve work together, so we can identify pathways by which demyelination can occur or axonal degeneration can occur. So then we can try to manipulate those pathways and make rational treatments rather than just guesswork treatments. So just in this sense, the new number of genes is really exciting. But what I thought I would focus on is just some of the research that's going on in a few of the more common forms of CMT, just to try to give you a picture of how this is working in the laboratory. And then I also want to show how that people like Mary and myself and others are working together to put the infrastructure in so that we can actually test whether or not candidate treatments that come out of the science actually are effective. And again, that's going to be the focus of my talk. And because one of the roles I play is I'm involved with the American version of CMT UK, which is called the Charcot-Marie-Tooth Association. And I'm going to talk about some projects that are part of what's called their STAR initiative, which is the strategy to accelerate research. And what the CMTA did was, rather than just accept applications from individual researchers, it sort of put together a team of scientists with expertise in myelin biology or axonal biology or clinical management of CMT, and have them get together and figure out ways to try to focus research in these diseases, to try to really focus on trying to bring therapies into fruition. And I'll show you some examples of that. So the first disorder I'm going to discuss is the most common type of CMT, which affects about half of everyone who has CMT, and most of the people who have CMT1, which is a demyelinating form of CMT. And this is CMT1A, and it's caused by a duplication of a piece of DNA on chromosome 17, and this duplication contains a gene which is named by where it is. So it's called peripheral myelin protein 22, and the 22 refers to its size, and because we're too lazy to say peripheral myelin protein all the time, we just abbreviate it as PMP22. And people, as you may know, with CMT1A have an extra copy of the gene for PMP22, so they make too much of it, and that causes CMT1A. And this is a result of what we call a dosage problem, and I'll show you just a little bit of information to support that. So this I'm very proud of because this is my skin. So this is a skin biopsy taken from this finger or this finger, I can't remember because I've had a lot of them done. And what it shows is that if you, I don't know if you can see this well here, but if you look up here in the top panel, those little black rings in that blue, those are myelin sheaths. And so you can actually take a skin biopsy and you can look at myelin just from the nerves there. So this is a much more benign procedure than doing a serum nerve biopsy. And if you look over here, you can actually see the myelinated nerve fibers going up into the skin. And what you can do is you can do some special techniques on even electron microscopy from those figures. And that's what this is. It's called immuno-EM. And you get what are called antibodies that recognize the PMP22 protein. You can couple them with little gold particles, and they'll label the PMP22 in the nerve. And you can actually see how much PMP22 there is. And if you look over here on this side, you can see in people, each one of those little dots is somebody with CMT1A. And if you look at people with CMT1A, you see they have a lot. People without CMT are in the middle. And people with a disorder called HMPP, where you only have one copy of the PMP22 gene, have just a little bit. So the disease you get, it correlates with how much PMP22 is expressed. And the job of the CMTA was to try to see if there was a way to identify compounds that can reduce the amount of PMP22 in the nerves. So are you with me so far, or am I off in left field? Or someplace in the middle as usual here. So one way historically that you could look for compounds that would reduce PMP22 was if people had an idea in a lab and they thought that this gene and protein might be a good way to treat CMT1A, you could see if you could manipulate that in a mouse model. And that's sort of like over here on the left. And we did that for the CMT1A trial, which finished a couple years ago. And it took about 3 to 5 years to get all of that done. And it turned out not to work. And we could have gone candidate medication by candidate medication. And with that approach, maybe in about 200 years that we might have found something. And I think that's a little long for most people in this room. And I was not planning on working for another 200 years. So. Alternatively, you could have used other organisms to test candidate medications. So these are worm models, and you can do what are called zebrafish models, and you can test more and more compounds doing that way. But if you had cells in a dish that you could use over here with a technology called high throughput screens, then you could look at actually thousands and thousands of potential medications that might already be out there that would reduce the levels of PMP22. So that's the approach that the CMTA decided to do, and I'll show you what's been happening with that so far. So if you're going to take that approach, you needed to have two things to do high throughput screens. One, you had to have good cells that represented Schwann cells, because those are the cells that are affected by CMT1A. And you had to have cells that always behaved exactly the same way in a culture dish. Now, they could have asked me to make these cells, but in the laboratory, I'm famous for dropping things on the floor, messing up my neighbor's experiments, and so that wouldn't have been a good choice. So what the CMTA did was they looked to find people who really were the most expert people in developing these cell lines. And the first person is sort of blacked out here, but that's Uli Suter from Switzerland, who was the person who first discovered PMP22. And the person on top is a guy named John Severin from the University of Wisconsin. Maybe I'll just get a little bit of water here so I don't croak here completely. Thanks. And John is the world expert on how the levels of PMP22 get regulated in the cells. So they were chosen to develop the cell lines. And this is a complicated slide, but I'm going to show you what John has done here, and hopefully it can make sense. So on top here is like one of the few art forms that I can actually do, which is called a straight line. And this straight line is a way that we can draw a gene. So I don't have time to go into too much basic information, so I'm just going to say this very briefly. But a gene is a piece of DNA that makes a protein, and it's specific for that particular protein. So what I'm showing you here is the gene for PMP22. And the way that the DNA works is that there's boxes up here, like let's name 1, 2, 3, 4, and 5. And those are the parts of the DNA that actually make the protein. Much of the other material helps regulate how much of that protein gets made in cells. So in Schwann cells, a lot of PMP22 gets made. In other cell types, hardly any PMP22 gets made. So PMP22 is expressed mainly in myelinating Schwann cells of the peripheral nervous system, which is why people get a peripheral neuropathy when they have the duplication. And the reason it doesn't affect other organs is because there are regulatory regions of the DNA which don't let it get made in other cells. Okay with that? Okay. And what John found is that there's a region between what's called exon 3 and 4, which is really responsible for that regulation. And so this is sort of like the on-off switch for the gene, except that it has a dimmer, so you can figure out how much it makes. And what John did was he took this region, and then to be able to measure PMP22 by various techniques in a dish is cumbersome, and it requires a lot of studies to do. So John said, well, let's see if we can find a reporter that can make that easier. So we took this regulatory domain of PMP22, and you guys have fireflies in the Midlands, or because fireflies glow, and they glow because of a protein called luciferase, which glows green. And so John took that protein and the DNA for that protein and hooked it up to this regulatory domain. So now every time a cell makes PMP22, it will glow. And then what he did was we have to find a cell that looks like a Schwann cell, and he went back and he found cells which are called S16 cells, which were discovered at the NIH about 20 or 30 years ago. And he has previously looked, and this is a complicated slide here, but basically it shows that these cells make PMP22, and they make PMP22 at virtually the same level and under the same conditions that Schwann cells in the peripheral nervous system make PMP22. And they all behave the same way. And so John made this construct with cells that glow, and he put them into these S16 cells. So now you have a cell line in which the cells will glow based on how much PMP22 they make. And so if you had medications that would decrease the amount of PMP22, you would decrease the glowing in the cells. Okay? Now, the next thing is how to measure them in a way that you can look at thousands of materials. And again, if they asked me to do an assay, I would drop the plate on the floor. But there are people who have real expertise, and we went all over the United States to find what we thought would be the best place to look at this. And there's a facility at the NIH, and it's headed by a guy named Jim Inglesi. And the name at the time was the NIH Chemical Genomics Center. The name's changed, and I actually forget what they call it now, but it's still run by Jim. And the CMTA hired this guy, Sung Wook, here to work as a postdoc on the project. And I'll show you the system they have at the NIH for this. So these are computerized robots, and they're going to pull out plastic plates. And each of those plastic plates will have 1,500 wells, and the robots put exactly the same amount of those cells that John made in each well. And then other robots take medications from libraries that the NIH has or drug companies have of all their compounds to see are there any in there that will reduce the level of PMP22 and not make the cells sick. And that's what's happening here. I'll just let this play through for a second. So this was patterned after Detroit and the auto assembly plants, which since I used to live in Detroit, I always liked that. So again, this is the system in operation. And this is, again, it looks like a bit of a busy slide, but it's sort of a walkthrough of what happens at each step here. So on the top up here is when you take a library of different candidate medications and you put them through those cell lines. And the reporters tell you if there's a compound which reduces PMP22 expression and does not make the cells sick. Then, when if a candidate medication passes that, it goes to another test where this time, just to make sure it's not an artifact by the reporter or the glowing assay, then the actual amount of PMP22 before and after treatment with that medication is looked at. Then what happens is if it passes that, it gets put into an animal model, which I'll show you. And if it passes that, then it can be taken into clinical trials. And this, on the right part of the slide, is just one candidate medication which passed the screen but can't be used because of toxicity. But you can just see on the top here, this is the amount of this medication. And going up on the y-axis here is how much PMP22 level there was. And you can see that as you increase the concentration of this compound, it drops the amount of the luciferase activity, and it doesn't hurt the cells at that level. And then down here is just showing the protein assays. So that's the approach for PMP22. And where are we now? Well, the CMTA, because this approach has been put into place, is in a nice situation with industry because they've already developed an assay, and so now companies are interested in working with them to test their libraries. So in the last month, there's a partnership with Sanofi Genzyme, where Sanofi Genzyme has used this assay to screen 2 million compound medications. At the same time, there's a partnership with the NIH, and they have a library of 500,000 compounds which have been screened. So those screens have already been completed at the initial stage. There's candidates in both of them that are being worked on now. So I think the message that I hope comes out of this part of the talk is that this isn't a pipe dream to have candidate medications that are going to be out there to test in clinical trials for CMT1A. Those are taking place, and within the next couple of years, I'm sure we're going to have trials in CMT1A that will be a lot more exciting than the vitamin C trial was. And this is just to show that once medications come out of those screens and cells, they are tested in a rat and a mouse model of CMT1A, and these are the best models of these disorders that we know of. But I think another nice step has been made here to try to remove bias from the system. So the CMTA has taken both the rat and the mouse, and they've obtained them from the original investigators and have put them into a neutral facility, which is called Psychogenics, which is in New York, which exists just to test animal models for disease. So in the words of one of my colleagues, Larry Rabbits, this is sort of a mouse hospital where all they do is perform various ways to look carefully at whether mouse behavior is changed with candidates in which the person who created them, the mice, or who brought the drug in, has really no say in the outcome. It's non-biased observations. So I think that's a really nice setup to have in place, and again, that is in place. So that's CMT1A. The next disorder I'm going to talk about a little bit is one of the more common types of CMT1, which is called CMT1B. And CMT1B is caused by mutations in another myelin protein, which is called myelin protein 0. And unlike CMT1A, which was a duplication of a protein but no mutations in it, this is not caused by dosage. This is caused by changes introduced into the Schwann cell by one of these now up to 200 mutations. So just reducing the dose of myelin protein 0 is not going to fix the disease. You have to figure out how the mutations cause disease and try to address that path. And one path that's affected by many myelin protein 0 mutations was started, first detected by Larry Rabbits and his colleagues. And what happens in these patients is that the mutation disturbs the folding of the protein. So when the protein's made, it can't be taken and put into myelin. And it gets stuck in a part of the misfolded protein gets stuck in a part of the cell that's called the endoplasmic reticulum, or the ER. And when it gets stuck there, it activates a process, which I'm going to show you, which is called the unfolded protein response. And the reason it activates this, again, is the mutated protein can't fold right. With me, or am I in left field again? Okay. So this on top is the endoplasmic reticulum, and when the protein can't get folded right, it activates this unfolded protein response by one of three pathways: a PERK pathway, an IRE1 alpha pathway, or an ATF6 pathway. I'm just going to highlight these on the next slide here. That's the ATF6, the IRE1 alpha, and the PERK pathway. So when the protein can't get folded right, it causes these pathways to act in a way that changes the cell. So for the ATF6 pathway, it goes down here in the cytoplasm of the cell, the part that's not the nucleus of the cell, and it causes a change here, which goes down into the center of the cell where the nucleus is, that's where the DNA is, and causes a change there. The second pathway, which is an important one for us, causes a change in what's called XBP1. And it changes the sequence of XBP1 and enables it to go to the nucleus. And I'm going to come back to that point because we're able to manipulate this and use another reporter and get a green marker, and I'll show you that. And the final pathway is the PERK pathway, which also goes down towards the nucleus of the cell to change the behavior of the cell. And that can be changed, and that can be recognized by looking to be able to label this molecule called CHOP. And I'm going to show you how that can be used also to screen the effectiveness of treatments which alter this UPR pathway. So what we did was we got one of these reporters, like firefly luciferase, but this is called green fluorescent protein. And if that XBP1 goes into the nucleus to change the behavior of the cell and improve the neuropathy, you can see it like this. And you can do those skin biopsies. If you remember, I showed you the skin biopsies on somebody's finger. You can look, here's the peripheral nerves. This is a person without CMT, and here's a person with CMT, and you can see the red spots here. That's CHOP in the nucleus. So again, you can look in patients or in cells to see if UPR activation has occurred, and the CMTA has worked to develop assays which are now just about ready to run, looking to detect compounds that will activate this or will ameliorate this unfolded protein response, which is a way to approach treatments. Now, just today, there's one of the best scientific journals in the world. It's called Science, and there's a paper from a group in Oxford, along with Dr. Rabbits and his colleagues in Buffalo, who have identified another compound which can actually ultimately perhaps be used in clinical trials, which basically rescued the neuropathy from one of these UPR mutations in the mouse model of CMT1B. So I think, again, I don't want to get too complicated here, but the point I want to make is that CMT1B is another major type of CMT. It's one of the most common 4 forms. And again, there's rational treatments which are leading to clinical trials and have the ability to slow the progression of the disease. So again, I think that these are exciting times for here. Now I'm going to show you research from one other common form of CMT, which is the most frequent form of CMT type 2. And this is called CMT type 2A, and it's caused by a mutation in a gene called mitofusin 2, MFN2. And I think I have a slide to explain this coming up, but just as to say it even before I show the slide, MFN2 makes a protein that helps organelles in the cell, which are called mitochondria, fuse together. So mitochondria are the energy factories in cells, and in order to behave as a cell and do all the things that cells do, they need to have mitochondria in them to provide the energy. And to really work well, mitochondria have to stick together in clumps, which is called fusion, and then they have to break apart, which is called fission or fragmentation. And MFN2 is necessary for the mitochondria to fuse together, and in CMT2A, they can't fuse correctly, and that contributes to their disease. And when they can't fuse correctly, they can't be transported down the nerve to provide energy to the nerve. Okay, so that's the introduction to the disease. And I'm going to just show you this film clip because I probably don't need to show it for this audience, but I like to show it when I'm talking to doctors because not all CMT1A is very, very slowly progressive, and MFN2 patients can often be severely affected by the time a person is 20, as is the case with this young man. So again, CMT2A is often more aggressive than CMT1A, for example. And here's just another one of those straight lines, and it's just showing that on the top here, there are mutations that act on a domain called the GTPase domain, and that's where a lot of the mutations cluster. So how to fix this disease? So there's mutations in the MFN2 gene that disturb the normal function, perhaps by disrupting that GTPase domain. And simply reducing, trying to give more normal MFN2 doesn't fix that problem because the mutations screw up the functioning of other MFN2 in the cells. So there had to be a scientific approach to try to get at this problem to develop treatments. And it comes from this, which is called MFN1. So if you're a cell in the body, you have to fuse mitochondria together. And mitofusin 2 is a way many cells do it. But there's a backup plan in the cells, which is called mitofusin 1, which is similar but not identical to MFN2. And scientists have shown that if you increase the amount of MFN1 in a cell, that can overcome the problems with MFN2 mutations. So here's MFN1, and in nerve cells, there's usually less of that than there is of MFN2. In other cells, it's the reverse. So the strategy has been to try to develop or identify medications or compounds that can increase the amount of MFN1 in cells as a strategy to help treat this disease. And again, our friend John Severin has done the reverse of what he did with CMT1A. So in CMT1A, he tried to develop an assay to allow us to detect compounds that reduce PMP22. Here he's put reporters into the MFN1 gene to try to identify compounds which will increase MFN1 as a treatment approach for CMT2A. And these are his reporter constructs. The other thing that's been sort of lacking with this type of research is that we've gone straight from cell lines to animals to as part of our pathway to get to clinical trials. But there's been two problems with CMT2A. One is that the mouse models have been lousy for CMT2A. So when you put these mutations into mice, the mice just look at you and run around normally. They don't get a peripheral neuropathy at all. So you don't have a mouse model to look at the effectiveness of any candidate medication you might have developed for CMT2A. And also, if you go from like cell lines, there's no human factor anywhere in the equation. So what I'm first going to show you is work done by a trainee of Mary and myself, who's Mario Saporta. And what he did was, going back to these skin biopsies, he took the skin biopsies and grew up the skin cells, which are called fibroblasts, but then he turned them into stem cells that have the potential to become any type of cell you want, and those are called immortalized pluripotential stem cells, or iPSC cells, and then he turned those into nerve cells, and I'll just show you those. So he took a skin biopsy from a patient with this mutation here, the R364W. He grew up the skin fibroblasts and then treated them and made them stem cells. So here are the immortalized pluripotential stem cells from that patient. And then he drove those in to have them actually become nerve cells. So now these are nerve cells from a patient with that patient's mutation that can be looked at in screens for compounds. And this just shows that these nerves send out axons, which are sort of hard to see in this light, but that's that whole green line going out to the right. That's the nerve process, or the nerve axon. And these little dots here are the mitochondria, remember, at the heart of CMT2A. So he can look in assays at how these mitochondria move and fuse and behave. So now we have patient-derived cell lines for CMT2A, and we also have a new animal model which actually works, and this hasn't even been published yet. But there's been a lot of concern about the lack of an animal model for CMT2A. So what was done was using modern DNA genetic engineering, like made a rat model instead of a mouse model. And this rat model was made and now is being evaluated. And it also is at this place called Psychogenics in New York. And the NeuroCube is just one of the ways the animal is evaluated. And I'm not going to go through all the details, except to say that these mice have a clinical abnormalities that occur at about 2 or 3 months of age. They can be measured, and we have 2 different mutations with these. And Mary and I have another colleague that we work with named Stephen Scherer, who's just one of the best peripheral nerve people in the world. And Steve has looked under the microscope at these nerves, and they have an axonal neuropathy, which again can be looked at with treatment approaches. So I'm sorry if I wasn't clear enough in explaining the reason for those approaches I showed you, but as with CMT1B and CMT1A, I hope at least you can get the sense that these are real therapeutic approaches based on trying to understand the disease, and really are moving towards identifying candidate treatments for these types of CMT. And this kind of research is not just limited to the 3 types I showed you, but in the interest of time, I just focused on those. So my question to you then is, if medications are identified for different types of CMT, I want you to tell me how we would know if they work. But you're not allowed to answer, well, if all my CMT instantly goes away. So I'm interested in what people think. How can we tell if these medications work?
Trials.
I'm sorry?
Trials.
Okay, clinical trials. So clinical trials would mean that you get a bunch of patients with a particular disease process, give them the medications, and see how they do. And that would be the way you would determine if they're effective. So I agree with that, but I think what gets lost in here is the details. So we do the clinical trial, we give people the medicine. How do we know if the medicine is effective? Okay. And why do we need to do 2 groups? Why do we need a group with the disease and a group with like a placebo or a false medication. Oh, I mean, it is for comparison, but why do you think we need to do that? Well, so I would answer that with the first group of medications, if we completely cured CMT in a week, it would be obvious, right? But if we don't, we want to see if it's altering the progression of the disease, right? If it's slowing down the rate. And a lesson that we really learned in the vitamin C trial is that patients and doctors, everybody really gets into whether the medication is working, and actually both the treatment and the control group did better than the previous natural history group for CMT 1A. In other words, so to really be sure if there's a slowing of disease progression, you have to be able to compare it against people who don't have the medication, or else you'll get fooled. So that's a reason I think that you need a placebo control, and that's something that we can talk more about later. So that's part of what you need for a clinical trial, but what else do you need? Right, you need a way to measure it. I mean, if somebody couldn't walk and they can walk and they can jump, and then that's fine. But if it's slower than that, you have to have good outcome measures. And particularly when you're starting with medications which may have an effect of slowing the chronic disease rather than instantly reversing it, you really have to design these measures carefully. And I think that Mary Riley is one of the great clinical neurologists in the world. And I think Davide Persin from Milan is another one. And I've worked for a number of years trying to examine patients carefully. And the three of us together worked to develop what's called the CMT neuropathy score. And we talked about doing it together, and we all were as careful as possible. But then when we went to examine patients together, we found that there were all these inconsistencies with how one of us used a pin or where we put the tuning fork, just subtle things, but which were capable of creating differences in scores. And it even could depend upon if we both did it the same way but used different tuning forks. We got different values for vibration. So we have to be able to develop outcome measures that we do things the same way and that are sensitive. And I'm going to use that just to introduce the last part of my talk, because that's what all three of us and others have worked on, so that we are working together around the world to develop outcome measures and ways of looking at patients so we can measure progression and measure change in people with CMT. So this is what's called the Inherited Neuropathy Consortium, and this slide's a little out of date. We now have 17 sites from around the world, and I think I put up the wrong slide here, which I really apologize for this audience, because CMT UK is a part of this also. So we have sites ranging from Australia to the United States, in Europe, and we have involvement of patient advocacy groups, so the CMTA and CMT UK, we have Telethon in Italy, we have CMTA Australia, and as I said, we have CMT United Kingdom. And again, we work to develop these kind of measures. And these are some of the research projects that we do in this consortium. So over the last 5 years, we have enrolled over 7,000 participants into various natural history protocols and other protocols. And again, trying to develop outcome instruments, including instruments for measuring progression is one of the things that we really focus on. And I think I'm just going to show you some examples of that. So after the vitamin C trial, we realized that we weren't detecting enough change even in people without treatment in CMT1A for clinical trials. And that was with what was called the CMT neuropathy score that Mary, David, and I had worked to develop. So what we did was we made a new version of this. We changed it in a way to make it more sensitive. I'm not going to go through the details completely here, but this is a CMT neuropathy score. So if you see Mary in the clinic, if I see somebody in the States, this is what we use to quantify CMT in a person. It's a series of questions about their symptoms, things we find on the exam, and changes we see on the nerve conduction test. And the higher the score, the more problems somebody has, and the total is 36. And when we tested this together, as I said, Mary, David, and I noted differences that we worked to iron out, and to keep other people from doing that, in our consortium, every year or so we have meetings where we examine patients together, and some of you may have been part of that when we did that in London a couple of years ago. And for every site in the consortium, there are spoken directions that you have to ask for each component of this, and there's a film, which you may recognize Professor Riley here, and showing exactly where to put the tuning fork, exactly how to do this, and everybody has to be certified by going through this to be able to put clinical data in the consortium. Now, another thing that we've learned is that many of the changes in CMT occur within the first 2 decades of life. And the CMT neuropathy score requires cooperation, which is difficult for young children, and it's not so sensitive when you get under the age of 10 or even through puberty, because during growth, people are changing, and what's normal for a 6-year-old isn't normal for a 16-year-old. So with the help of Josh Burns from Sydney, who's a specialist in pediatric outcome design, and Rick Finkel from who's now at Orlando, Florida, we developed a pediatric system, which we also use, and which is also used at the London site. And this is just a busy picture, which just shows what's called the CMT pediatric scale. But the components of this, just to show you quickly, are we measure the age, the gender, the height, and weight, because this scale has to be normalized to a person's age and sex. There's a hand dexterity component. So these are functional tests that, unlike the CMT neuropathy score, this requires people to actually perform tasks. And again, you see some of these here. There is quantified grip strength, both at the ankles and in the hands. Quantified sensation tests. And then there's balance tests that require walking on this small beam on the floor or along a taped line here, with 1 foot, with eyes closed, eyes open. And again, you get a very quantified score up to a score of maximum 44. And then the nice thing that Josh did was he put a little calculator here, which is an electronic calculator. So you put all the measurements in, including the person's sex, height, and weight, and you click the icon here, and it normalizes it all for age. So again, we're really happy with this score. We're looking at this now several years out, and we're getting natural history data of how the CMT subtypes progress during children, which really will allow us to be able to look in clinical trials with pediatric studies. Now, how many people here think that their physicians really understand what it's like to have CMT and what's important in your lives because you have CMT? Mary excluded here.
I think the real issue for a lot of us is our GPs don't understand our condition.
Well,
our consultants like Mary certainly understand. Employees and understand that. Now, general practitioners don't.
And so I appreciate you saying that and giving the consultants like Mary and maybe even myself some sense of what it's like to have CMT, but in fact we don't have it. And the outcome measures that I've showed you so far have been designed by people like me, and so they're not patient-reported outcome measures. So they're based on things that we can measure, whether it's where you feel a pinprick or how you do with items for dexterity, but they're not really directed at what's important in people's lives. So as we move towards clinical trials, we're really working hard to design patient-reported outcome measures and quality of life measures that aren't standardized for all disease, but are specific for CMT. And again, Sindhu Ramchandran on the top and David Herman on the bottom have been at the forefront of that. And we have disability indexes, we have quality of life measures for adults and children that again have been designed through interactions with patients. And I don't know how many here are involved in our patient contact registry for our consortium, but if you go under the RDCRN for the Rare Disease Clinical Research Network, you can sign up for a patient contact registry, and through that we go back and forth with questionnaires eliciting opinions from people who have CMT that are really at the heart of designing all of these. Then again, this is not consortium work that I'm talking about, but this, I couldn't not mention some of the biomarker stuff, which has been actually designed at the National Hospital by Mary and her colleagues in the MRC Center. Because one of the exciting things that's coming up is their efforts to look at imaging, things like MRI as a way to measure change. And I think their work has the potential to change how we measure progression in CMT. And so that is going to change the whole world of these clinical trials. And then the final thing I want to just put up here is the, again, this patient contact registry, which is this, this is the link to it here, because this allows us to get input from people who have CMT as we go forward with clinical trials to make sure that when we see if a medication is effective or not effective, that we're not just saying it's effective because our measurements change, but because it has an effect on the quality of life for people who have CMT. And again, this patient contact registry, we have people from virtually all over the world who contribute to this, and this is really essential for us to do our job, which is to make sure that when we report improvement or lack of improvement, we're looking at things, items which are important to people who have CMT. So please consider joining this.
We have a link on our website.
Okay. And finally, we're proud of our consortium, but we realize it doesn't reach out everywhere. So using the same outcome measures, we have a CMT International database where people from various countries can join and look at people and evaluate people the same way that we do within our consortium. And then we're able to compare results basically around the world. So I want to try to say this right. Our goal is not to try to control the way the world evaluates people with CMT. Our goal is to try to make sure that people throughout the world can evaluate patients the same way so we can learn from each other and share with each other and identify the rare genes and see, in particular when we do clinical trials in the rare forms, to make sure that we can get as many people involved as possible. And I think that's the spirit behind this. And these are just a list of many of the people who have been involved in the work. And none of this can happen without involvement with you. I mean, we can do all the thinking and designing that we want, but if it's not a partnership with people who have CMT, it just won't work, because we have to see if medications are effective, we have to know how different types of CMT affect people, and again, it's really, this is a partnership between people and family with CMT and investigators like Mary and myself, and I greatly thank you for this, but I also want to make a little bit of a plea that all of what we've been talking about in terms of clinical trials and outcome measures depend upon being able to detect change over time. So if you go to a site involved here once, it's great, but we really need those follow-up visits, because without being able to measure how these instruments progress over time in people without treatment, it really hurts us in trying to design trials and interpret them. So again, I thank you for inviting me, and I'm happy to take questions or do whatever I'm told.
Well, folks, we've got about 5 minutes if anybody would like to ask Professor Shah a question, but I think it might be helpful if you wait for the microphone to arrive, because otherwise other folks can't hear what you're asking. So Tim is about to arrive with the microphone, and the first hand up is just here. Hand up, please.
Please. Sorry.
I was just intrigued that you have
Hold the microphone right close to your mouth, please.
You have the outcome measure, and the first part of the outcome measure, there was actually technical parts of it, and then it said weakness above the arm, and I just wondered why there was the difference. Is it because you're not interested about the weakness above the arm? Is it if it's not so important?
I'm not sure what you mean. You mean by your head and neck, or by?
No, there was actually different parts in how you measure.
Yes. So
someone's ability.
Yes.
Or someone's level of CMT.
So CMT in general, so the question is, why is there some measures just for this part of the arm as opposed to this part of the arm?
The parts of the outcome measure were more technical in the lower arm than they were the higher arm.
Yes, and so the reason is that most types of CMT present in what we call a length-dependent fashion. Okay, so it means that usually there's problems with your feet, and then a little bit later problems with the hands, and not in everybody, but in most people, it doesn't involve the upper leg, or in day-to-day activities, it doesn't involve the upper arm so much. So the scales are designed with that in mind. So if you have no problems with the arm, for example, you get a 0 for that.
Yeah.
A 0 is what you want to get with no disability. Then you look at changes in the hand, and since that's where most of the upper extremities will be, your scores will initially focus on whether there's any weakness in intrinsic hand muscles, and then if there is weakness, the score will then move up to the wrist, then here, and then the upper arm. So most people don't have a lot of findings on exam with the upper arm. Most people have some findings with the hands. So to be able to discriminate over time, that's why a lot of the focus is at the hand, and the same is true with the feet in the lower extremity.
Okay, thank you.
Okay.
Just really quickly, that would you ever consider having like a function outcome measure? So not looking so much at sort of the medical model, but more about what people can do.
Yes, and that's one of the things that, again, focusing on what's important to people who have CMT. And Mary and I are involved with a group in Holland who have developed a big questionnaire, which is sort of based on functional scoring. And so that's one of the other ways we're trying to look at that. And we really, I don't think we're satisfied that we have the most sensitive way to measure change in CMT. And what we're really doing in our consortium, because we've just probably going to be renewed for the next number of years, is to really look and compare those to really address your question, because it's an important one.
Just got time for one more question if anybody has one.
One of the things I found very interesting in one of your earlier slides was that when you looked at the amount of PMP22 being produced by some patients with CMT and compared it with the control group, there seemed to be a little bit of overlap.
No, it's a very perceptive question.
And I'm just wondering whether that correlates in any way with the severity of the illness, and whether you've got any explanations for why some people seem to be producing PMP in the normal range.
So it's a very perceptive question. And so the answer to the first part of it is, so there is variability in the amount of PMP22 from those skin biopsies, which are from just small areas, in some people compared to others. We did not see that variability in the control group. We did not see that variability in the HMPP group. So I think my sense is that that's a real finding. The second part of your question is, it did not correlate with the CMT neuropathy score in its first version. The significance of it is, this is just me, I was going to say hypothesizing. I think that's a euphemism for bullshitting here. But I think what is likely to be happening is when you make too much of the PMP22 gene, it throws off the stoichiometry or the mixing ratios of what's there to make normal myelin. And so even if it's not, the amount's not the same when you look at that skin biopsy, I think that the myelin recipe is thrown off enough, sort of like using too many eggs in a cake mix or things like that. And I think trying to understand how that sort of relationship in the mixing works to get the myelin in place is really at the heart of that. But I think it's an important observation.
Thank you.
Sure.